Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Before we begin, a note from our sponsor. I'm Richard Jacobs, Executive Director of the nonprofit Finding Genius Foundation and host of the Finding Genius Podcast. In late 2016, I was rear-ended at 65 miles an hour by a truck on the highway, which sent me off-road into a ditch. The impact of the collision gave me a concussion and other injuries. At the hospital, a CT scan showed that I had thyroid nodules, which turned out to be cancer. It was then, when I had a biopsy of my neck, that I realized even if I was a millionaire, I wouldn't want a second or a third biopsy due to the pain and the invasiveness of it. And appointments at that time for thyroid experts were 3 to 6 months out. And I was worried about dying now, even if that was irrational. So because of this, I've decided to raise money to conduct a literature review on steroids, on the causes of anxiety and depression, a condition that affects well over 50 million people in the United States and hundreds of millions worldwide. Our goal is to create a codex, a guide that reveals all possible treatments for anxiety and depression for people that live with the condition, or for loved ones that have it, as my wife and my son do. To find out more about our fundraiser, visit findinggeniusfoundation.org and click on current initiatives. And now, to our guest.
Hello, this is Richard Jacobs with the Finding Genius Podcast, now part of the Finding Genius Foundation. I have Danny Welch. We're going to talk about mitochondria in our cells and how they interact with cancer and metastases. So, Danny, thanks for coming.
Thank you, Rich.
Tell me what your position is now.
Yeah, I'm a professor of cancer biology at the University of Kansas Medical Center and Associate Director for Education and Career Development at the Cancer Center as well.
Okay, excellent. And tell me about your current research. What are you looking at?
Well, my lab focuses on the topic of metastasis, which is the spread of cancer to other tissues in the body where they'll grow to make a secondary tumor. And most of my career has been in looking at the genetics that underlie that process. We first started out looking for suppressing genes, like the brakes on a car that keep the cancers localized. And my lab, I can say, with talented people around me, has discovered 8 of those. And we've been studying their function. The first of them we named somewhat in fun, but it was called KISS-1 because we discovered it in Hershey, Pennsylvania when I was at Penn State. And KISS-1 ultimately turns out to be the master regulator of puberty. It's a neurotransmitter, and a patient cannot undergo puberty without KISS-1. So the irony of puberty beginning with a kiss shouldn't be lost on anybody listening.
Quick question here. How do you believe metastasis starts, and why does it start? In brain cancers, I've heard that certain kind of brain cancers don't seem to metastasize, but many other cancers do. So why do you think it happens in the first place, and what would be the mechanism?
That's a great question for which I don't have a really solid answer. Personally, my experience tells me that a lot of what cancer cells do is try and recapitulate what happened during embryogenesis or fetal development, and they're looking to find that partner to make them stop. So that's what I think is going on, but that's 100% speculation.
No, speculation is, I like it. It's okay.
And your question or your comment about brain cancers, they are really highly invasive, but they don't metastasize much. So that's also one of the key points when talking about the process of metastasis, is that people have to distinguish between just merely invading and going somewhere and colonizing another tissue.
If it does that, is it still considered cancer, or it's not a metastasis, but it spreads out, I guess, from the primary, and so it's
a cancer. Okay. So if they invade, that defines them as a cancer. But metastasis is a subset of those invasive cells. And anyway, the point I was going to make with the KISS-1 a minute ago was that KISS-1 is it allows every step except growth after the cells have already seeded other tissues. So we're really excited that could be a therapeutic for patients, because most of the earlier steps have taken place by the time a cancer's been diagnosed. So that's one of our exciting directions.
The role of KISS1 in normal tissue is what? And then what is the role of KISS1 in cancer?
So in normal tissue, it has a couple of different roles, but the major one that's been studied is it's made in a part of the brain that stimulates the pituitary axis and then the gonads to initiate puberty. And it also seems to regulate aspects of pregnancy. And there are a lot of labs that are studying that around the world who would be, I think, of interest to your audience. And in terms of the cancer, again, it's ambiguous what it's doing, but the cancer cells moved all these other tissues. And what KISS does is it sort of keeps them there in a state of suspended animation, a dormant state for a period of time.
Okay.
So basically my thinking is, if we can do that, then we rendered cancer controllable, a lot like diabetes. We don't cure diabetes, we just control it. And we can make cancer a chronic disease rather than a deadly acute one.
What would you do? You would downregulate the expression of KISS1 in cancer tumors?
Actually, we would administer KISS1 to a cancer patient. And the reason that's promising is it can be found in high levels in the bloodstream and have no negative effect on patients. It's currently used in clinical trials to help children who don't undergo puberty because of a defect in the KISS axis. So it's already proven to be safe in people, but understanding how it works in the cancer is really a direction that we'd like to go. So one of the things that popped up with KISS that I think was the reason that this call was originally scheduled is we had found that KISS1 regulates the reproduction or the genesis of mitochondria. And so it alters metabolism in cells. And that observation made us think a bit more about the possible roles of mitochondria in the process of metastasis. And a friend of mine, Kent Hunter, who's at the National Cancer Institute, did a really awesome experiment. He took a transgenic mouse that has an oncogene in it that would make mammary tumors or breast tumors in a mouse. And he crossed those with a bunch of other strains of mice and then looked in the kids and showed that some background genetics would suppress metastasis and others made the cancers more metastatic.
When you say more metastatic, what does that mean? They did it faster, they did it more, they went to different tissues they don't normally go to. Like, how did you characterize that?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Great question. Usually in mice, we don't see too many metastases outside of the lung in these models. So and then we confirmed it in our models. It made them go to more often to tissues, and then it allowed them to grow at different rates in different tissues. So it depending upon the tumor type and what was driving it, it made those changes. That was a vague answer, but I hope it made sense.
Yeah, no. It seems like cancer has a tropism for certain tissues. Like I looked up, common metastatic sites for various cancers. I can't tell. It doesn't seem to be clear enough signal, but I can't tell if certain cancers have certain tropisms for certain tissue, meaning like, this kind of breast cancer tends to go to the liver. If that's the case, why would that happen? Or do you think it's more of just a random process where metastatic sites tend to go?
It's not random. That's been known for a couple hundred years now. There are two lines of thinking, and I'll tell you a bit of both of them. One is and this is true for the most part, the cancers metastasize to the first capillary bed they encounter after they leave the primary tumor. So that explains a lot of colon cancer going to the liver or pancreas cancer going to the liver and the like. But then there are other cancers, and breast and prostate are among them, that go to most commonly the bone. And that's definitely not the first capillary bed they encounter. So that prompted a fellow named Stephen Paget back in the 1880s, almost 1880, 1890. He called it the seed in the soil, where the cancer cell is the seed and each tissue is a different soil. And you have to have a right match of the seed in the soil to get them to grow. And there's evidence that supports both of those theories. And I find it fascinating. Different labs have looked for vascular zip codes, they're commonly called. These are markers that are in each tissue, and the cancer cells recognize the right zip code, and they say, aha, this is a place with good soil for me.
How would cancer cells know that? Like, is a tumor releasing, extracellular vesicles that are doing niche construction? Or, how do you think that this happens?
They are doing that, but also almost every cell type has receptors on it that recognize things that are expressed on other molecules or surfaces. So they're just sticky fingers, basically. And the pattern of what those receptors varies by cell type. One of your points, you were asking, like, does a certain kind of breast cancer go to a certain tissue and that type of thing? A real kind of easy way to illustrate that is the most common breast cancers express the hormone receptors for progesterone and estrogen. And those tend to metastasize slower, but they go a lot to the bone. Then there's the triple negative breast cancer, which has none of the currently targetable growth factor receptors on them. And those metastasize early, but they tend to be more broad in their pattern of metastasis. So you were right in your question, and we still don't understand exactly how all that's happening.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Well, has anyone been able to capture a primary tumor in the process of starting metastasis? I guess, I've heard there's an epithelial to mesenchymal transition. How would you characterize how metastasis initially starts in a given tissue?
Well, you've mentioned the EMT, or epithelial mesenchymal transition. That is clearly an important aspect of many metastases, but there have been a few studies that show that that's not a requirement. In other words, there are many roads that lead to Rome, consider metastasis that way. So besides EMT, there are clusters of cells that will metastasize. I joke when I have some slides that I use when I'm teaching basic metastasis, and I refer to those clusters like teenagers at the local mall. They move as a pack, disrupting everything in their path. They don't necessarily mean harm, but they do it. So they can move as clusters. And another type of invasion towards metastasis involves amoeboid movement, where they're not even making proteinases that degrade the matrix that support stroma in the tissues. So Again, there are multiple ways to accomplish the same thing. And if you block one of them, but leave open the opportunity to do something else, it's still possible to get metastases. So from the perspective of when I mentioned studying suppressors of metastasis, that was experimentally a choice that we made, because if you block a step completely, the tumor cell cannot metastasize because it can't go to the next step. But if you promote something, but it's blocked at some other step, you would never know that it's promoted. I hope that's making some sense. So
but they would be like intermediate stages where, if you're able to characterize the metastasis process and look at, the different biochemical steps it goes through, then you wouldn't be able to have insight into that, right?
Yeah, I think we will get that insight, and we are getting that insight, but it's a bit more challenging because there are bypass mechanisms and the like. I liken it to a plate of spaghetti and meatballs at your favorite Italian restaurant. And we'll just use 2 meatballs, but you can put them at 9 and 3 on the clock. And all of those noodles are connecting points between the 2 meatballs. And experimentalists take a fork and they take one noodle and they twist it around their fork so it's a bite-sized piece. And then they say, well, this is the connection between those 2 meatballs, but the other meatballs and noodles, the meatballs are still there, and so are the other noodles. They just may not be as direct an effect.
Okay.
So,
who understands in the goriest detail how metastases start and proceed through completion? Like, what do you believe? We have a primary tumor hanging out there. Is it trying to metastasize the entire time it's around? Or does it reach critical mass and there's certain like quorum sensing or signaling that tells it, okay, we need to start finding new places to go to? Like, how do you think it happens?
I think there's evidence on both sides for that. If you look clinically at following tumors over a period of time, establishment of metastases from a clinician's perspective tends to occur late. The tumor's got to have a critical mass, it gets to a size, and the probability of finding metastases is higher. But in the last 2 or 3 years, with the advent of really sensitive techniques with PCR and the like, the field has begun to detect disseminating tumor cells well before a primary tumor can be detected by most imaging methods, which is darn frightening to think that they're already shedding cells so long before we know the cancer's even there.
So I guess moving to an area that you'll know more about, what is the role of mitochondria specifically in either tumor formation, the inception of cancer, or metastases?
I'm going to sidestep that slightly because there is almost no data that mitochondria are involved in the development of cancer by driving it or forcing it to happen. But what we think is really happening is that the genome and the mitochondria are modifying loci. So that experiment I started to talk about with Kent Hunter, where he did those crosses, it was always with female mice of the modifying strain. And since you inherit mitochondria only from your mother, the alternative explanation for his data was that the mitochondrial genome could be impacting the cancer to change its metastatic phenotype. So what we did is we made a mouse or set of mice that have the same nuclear genome, but we've given them different mitochondrial genomes. And by changing only the mitochondrial genetics, we could replicate the results from Kent Hunter's really elegant genetic crosses. So now we've isolated the mitochondrial genetics as a variable in the experiment. Doing that, we and I will say, when I called Kent and told him this, he did like any good scientist. He goes, I really have a hard time believing it, but I cannot deny the data. And that's, I appreciated that response because that's what we should all do when looking at any data. Okay, if the data experiment was done right, we've done the controls, the data are the data. Now we have to revise our hypothesis. Right. And he's been terrific in that way. And we're trying to look at ways to explore it together. So we replicated with the genetic crosses, but I think one of the fun things that come to a couple of points you've raised and asking questions. One morning, literally, I was taking a shower and kind of my brain was beginning to function. And I realized that when we do those crosses, it's not only the tumor cells that inherit the mitochondria, it's all the supporting stroma. So I was talking to my grad student, Amanda Brinker, and we said, well, we can test that. We can take tumor cells that are the same nuclear genetic background, and we can implant them into the mice that have only the mitochondria different in the stroma. And when we did that, we saw that the stroma would give a 3 to 5-fold difference in metastasis depending upon polymorphisms in the mitochondrial genome. 3 to 5 folds. That's just huge. We would take tumor cells and we injected them into these mice that have the mitochondria different in the stroma. That's all the tissue that supports the tumor. And
the structural tissue that supports the tumor is that called the stroma? Is that what you mean?
Yes.
Okay.
So when we did that, depending upon the mitochondria that were in the stroma, I'll give a detail. It just adds some clarity to it. A mouse called C57 that inhibited metastasis by 60 to 80%. When we have C3H mitochondria, that's another strain of mouse, metastasis was 3 or 5-fold more number of metastases.
So how long after you would inject? So these tumor cells that you're injecting, it's just the nuclear DNA part has been taken from exogenous tumor cells, and that's what's injected into the mouse, or is it wholesale cells that what? I mean, how have they been modified?
The tumor cells are just their cell lines that were isolated from mice that matched the nuclear genome of these. So that way we could eliminate the immune system as tissue transplant rejection as an explanation.
Okay.
So a couple of things that we've been looking at, and I just said we eliminated the immune system, but we didn't. We eliminated transplant immunity as a mechanism, but we found that the immune system is different in these mice, and the mitochondria control that. And virtually all immune cell killing involves a burst of activity from mitochondria. So that's why we started to look there. But since you mentioned quorum sensing, I'll tell you something that's as yet unpublished, but we're very excited about it. The prevailing theory nowadays about mitochondria is that they were ancient bacteria that were engulfed by some other cell.
Right.
And they've learned to coexist. So your comment of quorum sensing made me ask the question, if mitochondria are ancient bacteria, do they retain the ability to communicate with bacteria?
Yeah, I would suppose so. Or maybe, who knows?
And if they can communicate with bacteria, do they make things that are like quorum sensing that can alter the growth of bacteria? And if they do that, then the microbiome would change.
So I interviewed a woman named Florencia McAllister who's studying pancreatic cancer and pancreatic tumors, and she had told me, this is maybe 2 years ago, the localized microbiome around the tumors is different from the rest of the healthy pancreas. So it definitely seems like, once a tumor forms, there's a whole microenvironment there. I mean, it accumulates its own microbiome, et cetera, et cetera, et cetera. So it does seem like all that happens for sure.
Yeah. And so we said, let's just test this in a simpler system, because people studying microbiota in individual tissues, it's $100,000 experiment to do it in one tissue. So we couldn't do it that much, but we did it in just the feces, which have bucket loads of bacteria. And in fact, the mitochondria changed only between 8 and 15 bacteria in the microbiome, and it was very reproducible. Some grew more, some grew much less. And we are actively searching for what the signal is from the mitochondria that regulate those specific species of bacteria. So keep your fingers crossed for me, please. I'm excited by that because I think that opens up an entire new field to explain how the microbiome is influencing so much of what our body does. For example, in the context of mitochondria, people who are obese have different metabolisms than people who are non-obese. And microbiomes are emerging as one of the key regulators those things. So the bacteria are also communicating back to the mitochondria. So we're looking at some crosstalk there, and if we can just understand it, I think we're going to be in a much better place to try and deal with these complex kind of diseases.
Is cancer a separate life form? At what point, when a tumor starts to grow, does it have enough voice, let's say, in biochemical signaling within a tissue where it seems like, again, a tumor perhaps, is very heterogeneous, but perhaps it does act as one, and perhaps it is essentially a separate life form at some point.
Yeah, I only modify the words slightly. It cannot exist without the patient or the host, but it definitely has a set of behaviors that it's controlling, and it is trying to exert influence on the rest of the body. Unfortunately, it does it in a destructive way. Yeah, more parasitic than symbiotic.
I guess because at some point, again, its growth is so fast that it has to defend itself. Which kind of signals to me that it could be its own life form. It tricks our own immune system, and there's angiogenesis, there's metastasis, and these all seem to be the hallmarks of possibly of a life form.
Yeah, but I'm not disagreeing with you. It's just the nuance of some of the words.
Yeah, I understand. No
problem. I can't disagree with you, and that's, I think, part of the reason it's been so difficult. It's almost like they've got a brain, but not quite. So just when you think you've got a handle on things, they've tricked us, like the immune system. That has freaked me out since the early days when I was studying immunology. It's just how the immune cells can be tricked into actually helping cancer cells invade and metastasize. Their job is to eliminate the cancer, but it doesn't. But they're tricked into doing something different.
What kind of picture is coming together for you through all your research of cancer? How is it? I don't know. Where do you see this going? Your research. In one hand, we're talking about big concepts. Is cancer alive? Why is there metastasis, et cetera? How do the mitochondria interact? And then in other hands, you're going to like very narrow molecular mechanisms. Like the KISS1, adding that to the mix to slow or stop metastasis, et cetera. So, you kind of go back and forth. You zoom in and out. It sounds like between, the very specific and the more general. But overall, what is your picture of cancer? How is it different from when you first started your research years ago?
Oh, great question. Well, when I started graduate school, I remember learning the statistic that no woman with stage 4 breast cancer was expected to live 5 years. And today, that number is about 50% live 5 years with stage 4 or metastatic disease. So it's changed a lot, not enough, but a lot. From a philosophical level, when I started graduate school and working in a lab, I wanted to do something that would directly help patients. And I was looking at things that were, could I hand this directly to a patient, like a baton in a race? But I became more enamored with looking at some of the fundamental things that we were really early in the pipeline to handing it off to the patient. And so that's why you're hearing kind of the big picture and the narrow picture. From my perspective, there are two things, and I think both of them are somewhat related. At least I hope that you'll be convinced that they're related. So I mentioned the KISS, and we're hoping to make fragments of KISS1 that could be administered to patients. And that sounds trivial, but it's proven to be a bit more difficult than we would like. The mitochondria project is a lot more difficult because if you think about the genetics of the mitochondria, it's almost all RNA that it's encoding, transfer RNAs and ribosomal RNAs. And we had to reconcile the specificity with altering metabolism and stuff, and concluded the metabolism was not the key, but the sequence of those RNAs are the key. And so we've been looking at a new class of signaling molecules from these transfer RNAs and ribosomal RNAs that get secreted, and they're found in the plasma, and they are signaling molecules that represent a class of signaling molecules that really were first discovered less than 10 years ago, called tRNA fragments and ribosomal RNA fragments.
Okay.
And right now we are trying to isolate, define, and reproduce those tRNA fragments where we have polymorphisms in the mitochondria and test their functions. And the fact that they are secreted into plasma and they can be found there in some clinical samples means they're stable enough that we could make drugs or drug-like molecules from them. So a goal for me in my career is just to have one thing, which would be more than most, that actually makes it into the clinic. But that's the long-term objective. The shorter-term stuff is to understand how both of those molecules are working. And even if it doesn't make it to the clinic as I'm outlining it here, hopefully it'll set the stage for the next person.
Since we've been talking about mitochondria, I totally forgot the Warburg effect in cancer. I mean, there's a lot of, the metabolic theory of cancer seems to say that mitochondria are damaged. I haven't heard explained how or why. In cancer, I mean, do you observe that? And so when you're doing experiments with mitochondria in cancer, how do you know that the material you're using is not damaged in such a way as to be skewed where you're not getting a true result?
It is interesting. The Warburg effect kind of languished for about 60 years and then suddenly has made a resurgence. The mitochondria don't appear to be all that damaged. They are just regulated differently. I'm going to, if you have never talked to Matthew Vanderheiden at MIT or Chi Van Dang, who's at Wistar, those two, and Craig Thompson at Sloan Kettering, those three could give you about 1 billion times better explanation of the Warburg effects.
Okay. Well, hopefully I can speak to them at some point soon.
Yeah,
I'll ask you later for referrals. But yeah, that's great.
So, but with regard to the way we made our mice, one of the things we did is we did not use traditional methods to make the mice and switch the mitochondria. We actually used a microneedle to move the nuclei. So they were never exposed to any mutagens, and we sequenced them so they are exactly normal as from the normal mice that would have just bred, except we swapped out the nucleus. So, but your question had an underlying point, and that's what we were trying to avoid. We didn't want to screw up the mitochondria so that was driving what we were studying. And as best we can tell, we didn't, but hopefully we didn't. But there's always a possibility there was some effect that we have not been made aware of.
In your research, though, do you have experts on mitochondria that you consult with or employ? Or
I talk to people at various institutions. I have a colleague, Isidore Ragoutsis at Thomas Jefferson, who's a leading expert in these fragments that I was talking about, tRNA and RNA fragments. Scott Ballinger, with whom we made the mice, he's studying these mice in the context of heart disease and diabetes, where they've had tremendous effects. So we talk all the time. I shouldn't admit this because it's going public, but I will go and say, for any student who might be hearing it, I hated studying all the metabolism stuff. And now I regret not being much more proficient in it, but I'm glad there are people who are, that I can draw upon. And we each bring a little different perspective to the story. And that's what I think makes our science kind of more interesting, because they see something in what I'm doing from a different perspective, but we're looking at the same thing, and we're both able to achieve from it.
Yeah, no, that's excellent. So, very good. So, I guess last question or so, what do you see as possible in the next 5 or 10 years based on your research? I
hope that we'll do a couple of things. One is with some of the metastasis suppressors that we'll be able to. I'm meeting with a colleague here we're hoping to design some targeting agents to carry them into cancer cells, and we'll try and test those. From the mitochondrial project, which I think I'm devoting more of my mental energy to it now, because to me it's so different that it excites me in a way that these suppressors we've been studying for 20 years, they're still exciting, but I kind of like new. So shiny object in the corner. I think these tRNA fragments, if I were in my 20s, I would think I was at the earliest stages of something that might be like CRISPR or microRNAs and that type of thing in terms of how they're going to change the field. And I've never said that kind of thing before, but this is so fundamental. They're present in bacteria, viruses. They regulate retrotransposons. The breadth that these things are going to impact is already being elucidated, but nobody knows how they're working. And I think that's going to be a 5 to 10 year eureka moment.
Okay.
I hope that's correct.
Yeah. Yeah, no, it makes sense.
So that's where we're headed. Hopefully we'll get there.
But Danny, you've been a great guest. I really appreciate you being here. And what's the best way for people to find out more about your work? Where can they go?
If they go to the kumc.edu, the Kansas University Medical Center.edu, there's a website there. I am not a big person on social media or anything like that, but if they reach out, if they have questions, feel free. There's ways to contact me via the website, and I will shamelessly listen to every idea and integrate it if I think I can, and hopefully reciprocate with some other kind of discussion.
Okay. Well, very good. Well, Danny, thank you so much for coming on the podcast. I appreciate it.
Thank you. I hope you have a great holiday season.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.